All right, so this is 5.2, which is mitosis, which what ties into the cell cycle that we learned about before, and differentiation, which we already kind of talked about when we mentioned how you turn into a multicellular organism with different tissue types when we were doing 1.2. So hopefully you remember that. So the three explorations that are going to be in your reading: chromosomes, mitosis and cytokinesis, and cell differentiation. All right, so our essential question is going to be, how does an egg sperm fertilization give rise to you? What are the steps and processes that allow that multicellular organism to arise from a single cell? Do all the somatic cells, that means body cells in your body, have the same DNA? Yes or no? What do you think? We kind of already talked about this, so you should know the answer. Kieran, Izzy, Liam, what do you think? All right, so most of you do remember that all the cells in your body do, in fact, have the same DNA. Um, because that cell, the zygote, undergoes mitosis. What does mitosis give you? Two genetically identical daughter cells. So you should remember, too, that DNA contains the recipes to make proteins. So that's what the sequence of nucleotides in DNA contains, the information to make, the instructions to make proteins. And you hopefully remember that proteins, which are a polymer, are made up of the monomer amino acids. Remember, each amino acid is different because of its R side chain. And those proteins are what cause everything to happen in living cells. So proteins do pretty much everything. They're part of the structure. Enzymes are proteins, and they complete most of the chemical reactions in cells. So normally, when a cell is just living, the DNA is lightly coiled. So hopefully you remember back when we did the coloring of the eukaryotic cell, you had that bowl of spaghetti in the middle of the cell, and that DNA was all spilled out. Looks like spaghetti. Um, that DNA is lightly coiled so that the information in the DNA can be read in order to make those proteins that that cell needs to live. So during interphase, which remember is G1, synthesis in G2, DNA is coiled around histone balls forming chromatin. I'll show you a picture in a second. The histones then coil even further, so they are in the nucleosome form, which is considered to be uncoiled. And then before mitosis, it becomes even more coiled up. Think of fishing wire. If you've ever had a big knot because it gets tangled, the DNA, if it didn't coil up in order for mitosis to occur, could get tangled and damaged. And think of a recipe that's never made. You don't need to have that bookmarked, right? It can even be kind of like clamped together in between other recipes that are frequently made. So not everything has to be easily read all the time. And I like this picture because it does look like spaghetti. So in the nucleus, when a cell is just living in interphase, you'll have this chromatin material so that the instructions for proteins that need to be made are accessible. Right? And it's in its chromatin form, it's wrapped around histones to form little nucleosomes. Now, when it gets ready to undergo mitosis, it condenses even further. Um, and I'll go over the different parts, but it's important to realize that this is so tightly coiled that none of these proteins can be made. None of this information can be accessed because it's too tightly coiled. Uh,
In this animation, we'll see a remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled in the chromosomes. In this
All right, so hopefully that helped you visualize that. What does DNA code for? In other words, what does the information contained in the DNA sequence actually give rise to? Kieran? Liam?
You're muted.
Kieran? You want to answer? Okay, good. Oh. So the correct answer for that was proteins. Why are genes that are used frequently only lightly coiled? So the correct answer for that is so that the gene that is used often can be accessed and read, and those proteins produced. All right, so here you see the DNA sequence that needs to be read. So that could make, consider that that makes a protein, and then it'll coil around those histones, and then when it gets time to undergo mitosis, you'll have further coiling into that chromosome characteristic shape. So when a cell approaches the mitotic stage, the DNA condenses quite tightly. So chromosomes are long strands of DNA, and this is where it can get a little confusing. You have chromosomes, chromatin. They all kind of sound similar, so you have to make sure that you understand the difference. But chromosomes can be used interchangeably for the entire length of DNA, even when it's in its chromatin form. It can be also when it's in its condensed form. And if you have a duplicated chromosome, that characteristic X shape that I'll show you in a second, that's also considered a chromosome. But chromosomes are long strands of DNA that contain the recipes to make thousands of proteins. And it gets replicated during the synthesis stage. And then in G2, when it's getting ready for mitosis, that's when it coils up. So here you see this characteristic X shape. That's not what chromosomes normally look like. It looks like spaghetti, and it only goes to this form when it's getting ready to divide. And the reason for that is you don't want the uncoiled form being pulled to either side to get put into each daughter cell, because you would have damage to the DNA as it's being pulled. The spindle fibers that pull the DNA to either daughter cell are actually the same chemical structure that your muscle cells are made out of. So they can be pretty, they're pulling on this material to get them to move, to get each sister chromatid to move. And if it was loose, the chance of damage would be greater. So here you see your DNA helix. It's wound around those histone balls, and then right before mitosis, it condenses to form that chromosome. What form is the DNA in when the cell is just about to divide? All right, so the correct answer is chromosome, and you can see the chromosome is right before division. Normally it's in the chromatin stage, and the DNA helix is what is around the histones. So one of the things, the cell doesn't want to stay in its chromosome form because all of those genes, those recipes for proteins, can't be read. So no new proteins can be produced, or we'll get into more detail. That's not quite 100% true. And one thing I want to just note, that each side of the X shape that we just saw is a duplicate of the other side. So whenever you see that characteristic X shape, those are considered sister chromatids because they're exactly the same DNA, right? You might say to yourself, but I thought I had one chromosome from mom and one chromosome from dad. Yes, and chromosome from dad will have a duplicate attached to it at the centromere, and chromosome from mom will have the duplicate, because when that cell goes to divide, it has to make sure that each daughter cell has the exact same DNA. So during the stage of interphase, that DNA is replicated. It's held to its sister chromatid by the centromere, and then when it goes to each side, it separates at the centromere. We'll look at chromosome structure later on, but where the centromere is in relation to the chromosome is indicative to which chromosome that actually is. They're ordered in, so number 1 is the longest one, number 2 is the second longest, etc. So the portion that holds each copy together is called the centromere. And at the end of each chromosome, on both sides, is something called a telomere. We have spoken about it a little bit in the last section. So telomeres are a portion of DNA that doesn't actually code for anything. It's not part of a gene or a recipe for a protein, because when when the DNA is replicated, the enzyme that actually replicates the DNA can't replicate those last few bases. So every time a cell undergoes mitosis, a few bases are lost. So after many, many rounds of mitosis, the telomere gets eroded, and then the cell starts losing information from the recipes for proteins. So that's why eukaryotic cells have those telomeres on the ends of their chromosomes, because the cellular machinery that replicates that DNA can't go to the very, very end. So every time you undergo mitosis, that chromosome is a little bit shorter. So if there's, buffer DNA at the end, it's not going to start eating into the important DNA, the genes, the recipes, until it's pretty old. So where is the centromere? Right, so 2 is the centromere, right? And um, remember that each one of these chromosomes is an exact replica that's created during the synthesis stage. Where is the telomere? All right, so 1 is a telomere, it's at the end. 4 is a little further up, but if you picked 4, that's good. You shouldn't have picked 2 because that's the centromere. All right, so just like I was talking about during mitosis, DNA polymerase, which is what makes the new DNA, and we'll get into that when we do the chemistry of DNA. Can't go to the end, so each subsequent division, the chromosome's a little bit shorter. So what's really interesting, some cancers actually add telomeres so that they can continue to divide to make bigger and better tumors. And some viruses also do this. So scientists are actually looking into how to add telomeres onto cells using these the methods that viruses and some cancers do. If that were the case, you would have the ability to never grow old physically. So if you could replenish the telomeres, you would stop aging at the age that you started replacing your telomeres. You'd never be able to go back to get the cells that had perished. But you wouldn't have any wrinkles, you wouldn't have any joint or skin or connective tissue issues. All your organs would be able to continue functioning without any decrease in their efficiency or their ability to perform their functions. All right, so just to kind of go back to what we're talking about in mitosis, remember that the cell cycle is gap1, synthesis gap2, that's interphase, and then mitosis and cytokinesis are the acts of cell division. And mitosis results in 2 genetically identical daughter cells. Okay. Now, most eukaryotes use sexual reproduction to reproduce themselves, and mitosis for repair, replacement, growth. Some eukaryotes, though, can use mitosis for reproduction, and these are limited to a few instances. This includes vegetative propagation, parthenogenesis, and budding. So the steps of mitosis, interphase is G1, synthesis G2. Then you have prophase, metaphase, anaphase, and telophase. And I'll go over those steps right now. Prophase is when the chromosomes condense into those chromosomes. Centrosomes, don't confuse that with the centriole, act as anchors to pull the DNA to each side so the cell can be split in the middle. Spindle fibers are the ropes that pull the DNA. Again, they're the same chemical structure as muscle cells. I know all these words kind of sound the same. Then you have metaphase, where, again, the spindle fibers are attached to the centrosome and the centromere. Centromere is the middle of the chromosome. And the chromosomes line up on the equator, or the middle. The equator is the middle. So one easy way to remember that metaphase is when they're lined up on the equator is they're in the middle, M, metaphase. Then anaphase is when the sister chromatids that were part of that X move to each side. So they're separating at the centromere and moving towards either pole. Remember, they're twins, so each side is going to get one exact same copy. So one way to remember that is that anaphase are moving away from the middle. A, anaphase, A, away. An interesting thing, if we were in person to do this lab, you'd have to look at a bunch of cells from an onion root tip, and during anaphase, the chromosomes actually look like an A without the middle. So that's one way you can remember anaphase. And then telophase is where the nucleus begins to reform, and the spindle fibers dissolve. But most importantly, the chromosomes begin to uncoil into chromatin so that they can be used to make their proteins. Then cytokinesis is where the cell actually physically divides. In in animal cells, you have like a little protein belt that kind of like tugs tightly and closes that off. And then in plant cells, you have a cell plate that forms. So then those two daughter cells enter into G1 of interphase, and the cell cycle begins again. So one way that I've come up with to help you remember the stages of mitosis as well as the order that they go in: pimples make all teens crazy, prophase, metaphase, anaphase, telophase, and cytokinesis. All right, what stage of mitosis has chromosomes lined up on the equator? Zach, Isaiah? All right, so lined up on the equator. Equator is the middle, so that's metaphase. What stage of mitosis has chromosomes moving away from the equator? Zach? All right, so the correct answer is anaphase. Telophase is when the nucleus is reforming and the chromosomes are uncoiling it back into the chromatin stage. All right, so like I had just mentioned before, in multicellular organisms, mitosis is usually used for growth, repair, replacement. We've talked about this already. Some multicellular eukaryotes use mitosis for reproduction. So this isn't usually the preferred way, because if you have an entire species of clones, what's going to happen if you have a disease or some type of environmental conditions that there's no variation for? If you have clones, that is leading to extinction for that species. What's up, David?
Because they're because they will all have the same flaw.
Well, it might not be a flaw, but if you have environmental conditions that are changing, right? So if you have a disease, they'll all be susceptible to that. But it is, in fact, used in some cases. So here are the examples where you have mitosis being used for reproduction. Budding, the most famous use is hydra. They're an aquatic species, and they'll grow another hydra off the side. Vegetative propagation, examples are potatoes. If you take an eye, for those of you in culinary, if you take an eye of a potato and put it in dirt with conditions that are favorable for growth, it will grow into a clone of the plant that produced the potato. If you have Miss Leal, you may have noticed she has that big spider plant in her room. And all those little spiders that are hanging off, if you put them in soil, they would grow into a clone of that plant. Fragmentation, the most common example are sea stars, right? Starfish. So if you cut an arm and part of the central disc off a starfish, it will grow into a clone of that original starfish. So it's kind of cool if you take a starfish and you cut off each arm and part of the central disc, you'll have 5 clones of the original starfish. Sponges, marine sponges also do this. Don't get this confused with a lizard dropping its tail, because that tail does not grow into a new lizard. So that's not an example of fragmentation. And the final one is parthenogenesis. Snakes and some reptiles will lay eggs that are clones of themselves. Um, Obviously, this is not favored. Sexual reproduction is favored. But in a case where an organism is isolated and can't find a mate very often, they will undergo parthenogenesis. Now, if you owned a snake, or you may have heard stories about people that have a snake, for 5-6 years, and all of a sudden it lays an egg, and there's baby snakes. So this can occur, and those babies are clones of the original the female, because they lay eggs. Now, prokaryotes can actually undergo mitosis, and we talked about this in the last section, because they're unicellular. So they do not have, prokaryotes have no nucleus, so you can't dissolve a nucleus, and you can't move chromosomes around when you don't have chromosomes, because they have a single loop of DNA. So prokaryotes undergo binary fission, and that's their way, akin to mitosis, to produce 2 genetically identical cells. So what happens in binary fission, the loop is copied. You don't have to worry about telomeres, because there's no end. And then the membrane and peptidoglycan cell wall, which is not like a plant cell wall, cleave off, and you have that new prokaryote. So it's very quick too, 20 minutes for bacterial reproduction. And this is just where you have a cleavage for binary fission, and it just cleaves into 2 new cells. And that's the loop of DNA. There is no nucleus in there. What do bacteria use for reproduction? Good, that's binary fission. What do only a few multicellular eukaryotes use for reproduction? All right, so only a few multicellular eukaryotes use mitosis, right? That's parthenogenesis, fragmentation, vegetative propagation, and budding. What do most multicellular eukaryotes use for reproduction? And that's meiosis. Meiosis is sexual reproduction, and we'll learn about that later. All right, so how do we get from that sperm egg fusion to the multicellular organism that you have organs and tissues and your cells do all kinds of interesting things? Despite the fact that we are all created, all those cells are created by mitosis. So we've kind of talked about cell differentiation way back at the beginning of the year. Remember that every single cell in your body has the same exact DNA because all of those were created by mitosis. The only cells that don't have the same DNA in your body are your egg and your sperm, and red blood cells because they don't have a nucleus, so they don't have their DNA still. So how does it happen? Remember, you start off as a zygote, which is a fertilized egg. This starts to divide via mitosis to form a blastula. So this is a hollow ball after 5 rounds of mitosis. Then you form that hollow ball has 3 different layers that I'll talk about in a second. And then the gastrula becomes more convoluted from the blastula. And those 3 layers are called germ layers of both the blastula and the gastrula have 3 germ layers, but it doesn't mean germ as in some microbe that can make you sick. It comes from the word germination. So those germ cells are able to germinate other cells. So the 3 layers are ectoderm, ecto meaning outer layer, and this gives rise to skin and neurons. The mesoderm or mesoderm is the middle layer, middle. This gives rise to bone, muscle, blood, kidneys, ligaments, tendons, connective tissue. And then the endoderm, inner layer, gives rise to your digestive organs, glands for the digestive organs, and parts of the respiratory and excretory systems. So I just wanted to kind of show you the difference between a blastula, which is just a round hollow sphere, and then your gastrula folds in on itself. In both cases, they have those 3 germ layers, the ectoderm, mesoderm, and endoderm. So the endoderm, again, forms, that's the inside, forms your lung, digestive cells and glands associated with that, such as the pancreas and thyroid cells. The ectoderm, which is the outside, forms the skin, pigment cells that make your skin colored, and neurons. So if you want to think of, the ectoderm, which is the external layer, and the external layer of you being your skin, hopefully that will help you remember. When in doubt, it's most likely the mesoderm, which is the middle layer, because you can see how many different tissues arise from that. Your muscle, bone, some kidney, blood cells, etc. All right, what layer of the gastrula will give rise to the skin? So here you can see that round formation, that ball of cells. This is already starting to become a gastrula. Isaiah, Izzy, Jace. All right, so remember your skin is on the outside of you. So what's that going to be? It's going to be the ectoderm, the outside. What layer of the gastrula will give rise to pancreatic cells? Hint, that's a gland involved in digestion. That's my hint. Gage, Lil, Zach. Good, that's the inside, the right, so these layers are made of stem cells, right? Stem cells, we've all heard. What does that mean? You can see moisturizer with plant stem cells. A stem cell is just a cell that can undergo mitosis many times and serve as like a factory or a manufacturer of particular types of cells. So these are cells that haven't committed to doing anything other than making more cells. Now there's different types of stem cells. Embryonic stem cells are the most powerful in terms of what they can turn into. Um, I'm going to go into the different types. Actually, let me just do it right now. So, totipotent stem cells come from the zygote before it gives rise to those three germ layers. So, up until like the fifth um, mitotic reproduction, all of those cells can turn into anything. So these can be made in a petri dish. If you have an egg, you can harvest eggs from women who are undergoing in vitro fertilization, fertilize it with a sperm, and then you have access to all these totipotent stem cells. Pluripotent is after those three germ layers have formed. They're still very powerful in that they can turn into any of those tissues that those the individual germ layer can turn into. Um, so they're still very useful, but they've already kind of been dedicated to a particular type. And then multipotent have differentiated even further. These occur when you have your fetus growing and you already have all that tissue development. And these can turn into only a few different types of um, other cells. So those totipotent cells that come from a zygote are the most useful. They're cell factories for any type of tissue. The reason that the U.S. doesn't do uh, stem cell research is very highly regulated. There's only a few different types of lineage. And the reason for that is because of some religious views that people have that um, any zygote, as soon as an egg and a sperm fertilize, that's a human, even if it's made in a petri dish. So that's why there are some pretty severe restrictions on stem cell research in the United States. This kind of puts us at a disadvantage because other countries have exceeded our uh, ability to use stem cells for different treatments. Um, there was also a big phase back in like the 90s and 2000s to harvest cord blood and then put it in storage in case someone's child had issues. Now, stem cells can only be used for that particular person. So when you see those moisturizers that say plant stem cells, they're useless. The stem cells have to come from the organism that they originated in because they need that name tag that they belong to you. So if I took stem cells from someone else's cord blood, they'd be useless. They would be attacked as if they were invaders. Um, Now, adult stem cells, because of CRISPR, which we're going to talk about later on in the year, um, you can take um, multipotent stem cells and then reverse them to pluripotent stem cells and then do different things with those stem cells. So you can kind of work with those stem cells and turn them into different tissue. And this is actually being done kind of in the US, but you have to fly out of the country and they'll harvest marrow cells and then use those stem cells in that to produce um, stem cells that they can inject into torn cartilage of the knee and then replace that or repair that tissue. So totipotent, no differentiation, can turn into any cell. Pluripotent from a blastula can turn into multiple systems, and then multipotent are the more restricted type. So this is just what I was saying, that you can have reverse engineer using CRISPR multipotent stem cells and turn them into pluripotent stem cells, and then actually use them for some treatment of injury or disease. So, one of the ways that these stem cells differentiate is by location. So, knowing who they are next to causes them to read certain recipes in the DNA to only access certain genes. And then other parts of DNA are kind of closed off when we talked about that. It's something called epigenetics, where you have material that kind of closes off that DNA so those genes aren't often made. But then sometimes cells that are very often used are kind of bookmarked. And that's how you get that differentiation. So when you have recipes that are used often because they're needed in life and are bookmarked, this is called epigenetics, and it's pretty poorly understood at this point. We're in the infancy of understanding the epigenetics. All right, so what is epigenetics? The bell just rang. Don't worry about answering that. And don't forget, the reading is going to be due tomorrow at midnight. So if you want to get started on that. All right, have a great day. Bye, guys.
Bye.